Hey guys, welcome to AP Biology, and this is our lecture on enzymes, energy, and metabolism. This picture I took back in 1993 showing a young baby gorilla, only less than a week old. I do want you to look at its face and ear, and the color. The color is light pink, and that is not typical of a gorilla. It's only found when the gorillas are very young because an enzyme has not yet been activated that actually leads to the production of melanin, which is the pigment that gives the dark coloration to their skin, as you see on his mother's chest right there. That's the normal color. So enzymes play roles in all sorts of things. And that's what we're going to talk about today are enzymes and what they do. So first of all bioenergetic and metabolic pathways are facilitated by enzymes. And enzymes are proteins. They are big proteins. So going through the 3D modeling, you learned about the enzymes and their active sites and the substrate molecules that fit into the active site in the enzyme. Once that happens, becomes the enzyme substrate complex. And this picture over here on these two pictures, I should say, are showing induced fit, where it's not a clock and key fit, but an induced fit, and the enzyme does sort of clamp around that substrate molecule, what you're looking at. So they are proteins. And we all know that proteins consist of these four levels of structure, or at least three of the four levels. They must have the three levels, but many proteins do have that fourth level. Primary protein structure, of course, is the sequence of amino acids, which is determined by your genes. That's what a gene is. The secondary protein structure is a result of a result of hydrogen bonds between the amine group and the carboxyl group of adjacent or nearby amino acids. And the tertiary protein structure is a properly folded polypeptide, and that may be the last level of structure for a particular enzyme. It might be a functional enzyme at that point, but most enzymes are really big, and so they consist of several of these tertiary structures put together to make the quaternary protein structure, such as we see at the end. And that's the enzyme catalase actually consists of four polypeptides that do come together to make the functional protein. So what enzymes do? They lower this thing we call the activation energy. What does that mean? Well, that means that we have to put a little bit of energy into the system in order for the reaction to occur. If this didn't happen, molecules would not stay together. They would just simply fly apart, okay? And let's look at this diagram. What do we have on the y-axis? We have free energy. What's that? That's the amount of energy that's available to do work. Okay, and we got progress of the reactions at the start of the reaction on the left-hand side, moving towards the end of the reaction on the right-hand side. And we can see that the reactants have more energy than the products. So energy is being released in this particular reaction, and this would be an example of a catabolic or exothermic reaction. So those are all terms you need to be able to link to a diagram such as this. This is a catabolic exothermic reaction, okay, that releases energy. And the black line represents the reaction, the amount of energy we would have to put into this system without the help of an enzyme. And the red line shows that with an enzyme, our activation energy that we have to put in is much lower, and therefore this reaction will happen at a faster rate and also happen at a lower energy input. And so overall, you will get more energy released out of an enzyme catalyzed reaction than out of a non-enzyme catalyzed reaction. I do also want to quickly say that enzymes will only catalyze reactions that will happen on their own without an enzyme. They just make the reactions go faster, a whole lot faster. Okay, so again, what do they do? We have on the bottom down here, we have the diagram we just looked at on top, and then we have a diagram, a little cartoon on the bottom showing what type of a reaction. This is a breaking reaction, so this would be a catabolic reaction, therefore an exothermic reaction, okay, showing how that happens. So what exactly do enzymes do? Well, let's look at this particular diagram. We have our substrates coming in, binding into the active site. Once again, we can see indication of induced fit. And once an enzyme is a substrate molecule is bound inside that enzyme, it's going to speed it up from a variety of different ways. It says over here under number 3, acting as a template for substrate orientation, meaning it's going to orientate those two substrates in a manner that's going to make breaking the bonds or creating the bonds more likely. It's going to stress and or stabilize the substrate molecules as they're going through what we call the transition state, whether that's a breaking catabolic reaction or an anabolic reaction. Oftentimes, they provide a favorable microenvironment because remember, the environment of most of these reactions is water. That's a polar environment. Well, the interior of the enzyme, the active site, might be a nonpolar environment. It also might be a charged environment, making it more likely for a particular reaction to occur. So those are all things that can happen there. And we all know that enzymes are proteins, therefore, they have optimal conditions. So, what are some of those optimal conditions that you got from your 3D enzyme modeling? All right, think. All right, you probably thought temperature and pH. Yay! All right, you got those correct. And I do want you to notice one thing about temperature. Okay, is that the rate of the reaction ramps up slowly with temperature. Why is that? Because at cold temperatures, enzymes are not really affected. At least their shape is not affected. What is affected is the movement of the molecules, the substrate and the enzymes. Okay, so if the substrate's moving around and the enzymes, let's say it's bound to a cell membrane or the internal membrane of a cell organelle or something of that nature, it's still random movement. And when temperatures heat up, that movement becomes faster, meaning that an enzyme substrate contact will be more likely to occur at warmer temperatures. But as soon as you hit the optimum temperature, we see a rapid drop off. Why? Because that temperature has caused the breaking of these weak bonds, the hydrogen bonds, in either the secondary structure or the tertiary structure of the protein, leading to a shape change, which changes its function. pH is a little bit different. pH at both higher and lower pH levels than the optimum, we see denaturation occurring there. And again, it is this secondary structure, which is all composed of hydrogen bonds between the amine and the carboxyl group of nearby / adjacent amino acids. These weak bonds are going to be the first ones to break. And when they break, that's going to cause a change in the tertiary structure. But there are also some hydrogen bonds, as you can see right here in the tertiary structure, that can also break. Now, if that pH change or the temperature change is not too drastic, then that protein may renature back to its normal shape once conditions return to normal. However, it's too extreme, it can lead to complete denaturation of that particular protein, and it will no longer ever function correctly. We learned about inhibitors during your 3D modeling also. The two types, we had competitive inhibition, where a competitor molecule actually competes for the active site and blocks the substrate molecule from coming in, or an allosteric or non-competitive inhibitor, which binds to a site other than the active site, but then causes a shape change in the active site that can cause or not allow substrate molecules to bind to that active site. So which one of these things is more detrimental to the enzyme? Or I don't want to say detrimental, but which one is more likely to shut down / slow down this reaction? We'll find out in a sec. But also, not only do we have allosteric inhibitors, like I mentioned here, or you saw in your 3D modeling, but there can also be allosteric activators. When the enzyme does not have the active site does not have the proper shape, as when the allosteric site is not bound by an activator, but once an activator binds to that allosteric site, it's going to cause a shape change in the active site that now makes it amenable for the substrate molecule to dock there, and then that goes and allows that particular reaction to occur. Many coenzymes and vitamins play a role as allosteric activators. Don't worry about this diagram. I got to refix that one. But this is an important graph. Oops, let's go backwards. Excuse me, guys. Shift. Okay, let's look at this graph real quickly. The green line represents a normal uninhibited enzyme reaction. This is the maximum rate. We can see increasing substrate concentration on the x-axis, and the enzyme's at a constant value. So what happens normally is as we increase substrate reaction, the rate goes up. But as we start to utilize all of the available enzymes, then the reaction rate begins to slow until we get to 100%. Every single enzyme is activated and it's going as fast as it can. Now, a competitive inhibitor um, slows down the reaction, but it does not shut it down. Because remember, it's competing for that active site, but if the competitive inhibitor is held at a constant value and we increase the substrate concentration, then the substrate begins to outnumber the competitive inhibitor and eventually it will reach its maximum speed. However, non-competitive inhibitors, which bind to a place other than the active site, alter the shape of the active site and therefore can shut that reaction down, as we see down here, very quickly. Now, this plays a huge role, these non-competitive inhibitors, because one of the things that they do is they help to save energy resources in an organism. Let's look at this metabolic pathway, which is where we're starting with the initial substrate of threonine, which is an amino acid, and we end up with the amino acid isoleucine. One thing you need to realize is that these metabolic pathways consist of many, many, many steps, each step catalyzed by a specific enzyme. So threonine comes in, and the first enzyme is enzyme 1-threonine deaminase, which is going to basically take off the amine group. And then we get intermediate A catalyzed by enzyme 2, which makes intermediate B, which is then catalyzed by enzyme 3, and so on until we end up with the end product, isoleucine. Well, in order to save energy, why continue to undergo this energy-intensive pathway if you have plenty of isoleucine available? So isoleucine acts as an allosteric inhibitor by binding to an allosteric site on enzyme 1, because we want to shut it down very quickly. We don't want to wait until we get to step intermediate D before we shut it down. We want to shut it down quickly. Okay, natural selection has led to that particular inactivation mode. And this enzyme will no longer function until isoleucine is actually running out and it is actually used up by the organism. And once it's used up by the organism, then the enzyme will reactivate because it no longer has isoleucine, causing a shape change in its active site. And then we begin to go ahead and make it. This is a form of negative feedback inhibition, okay, or feedback inhibition. You need to know about negative feedback inhibition. We're going to see this in a couple of other different examples. There's also positive feedback inhibition, and the most common example of positive feedback inhibition, or excuse me, positive feedback would be childbirth. As the baby is being born, its head is pushing against the cervix of the mother, and nerve endings there send a signal to the hypothalamus, which then signals the posterior pituitary gland to release a hormone called oxytocin. And oxytocin travels via the bloodstream to the uterus, which then causes muscle contractions. Which pushes the baby's head against the cervix, which then sends more signals to release more oxytocin, and it just keeps on getting more and more. The contractions get closer and closer until bing, out pops little Johnny. All right. And again, we want this process to happen relatively quickly, but it doesn't always happen as quick as we'd like to. But anyways, that is an example of positive feedback. We'll talk about some more later on too. There's a video here you can click on this and listen to. It's a great little recap of what enzymes, done by a friend of mine called, his name's Glenn Wokenfield. And there's also a short little animation of the video. Maybe we'll look at that in class. And that is it for enzymes.